Welcome back to Chapter 10. We're going to continue with Part 3. This is the last part of Chapter 10 that we'll be discussing. Some good compare and contrast between meiosis and mitosis is definitely necessary. So, mitosis is a conserving division. The number of chromosomes that end up happening at the end of mitosis is exactly the same as the number of chromosomes that were present at the beginning of mitosis. That's not true for meiosis. Meiosis reduces the number of chromosome sets from 2, or from being diploid, to being haploid. And that in and of itself produces cells that are genetically different from each other, as well as being genetically different from the parent cells. Meiosis has 2 divisions, and mitosis has 1 division. There are 3 extremely important events that happen in meiosis and only in meiosis, and all 3 of these happen during meiosis 1. Those 3 things, some of them are going to be very obvious to you, and some of them are going to be less obvious, and I'm going to go into some more detail about them. The first thing is that synapsis and crossing over happens, and we know that that happens during prophase 1. Remember, we've just gone into a lot of detail there in the previous part of this lecture. Homologous chromosomes are physically connected to each other, and they exchange genetic information. Something that's a little less obvious is that the way that those homologous chromosomes align on the metaphase plate during metaphase is random. So these chromosome pairs line up in random positions on that metaphase plate. I'm going to go into more details on that in subsequent slides here. And the final thing that happens that's unique to meiosis is that homologous chromosomes separate, and they separate during anaphase 1. So, we're going to finish doing that compare and contrast, and then it's actually over the next couple of slides, and then we're going to go into a few more details so that we can understand that very interesting thing with the alignment of the homologous pairs on the metaphase plate, so we can kind of get a better understanding of that. So, no matter what, mitosis or meiosis, sister chromatids do stay together. They are held together during called sister chromatid cohesion, but when that cohesion is interrupted varies. In mitosis, that cohesion is interrupted during anaphase, and in meiosis, that interruption is happening during anaphase 1. I'm sorry, the sister chromatid cohesion is interrupted during anaphase 2. Sorry about that. So again, let's just get a visual on this. If we look at mitosis, and notice that here we have 6 chromosomes, each of which has sister chromatids. So we say 2n equals 6. They go from being unreplicated to being replicated, and that happens during interphase for both mitosis and meiosis. But then, whereas in mitosis, you just kind of start lining up on the metaphase plate here, and it does not matter at all how they line up because sister chromatids are going to separate, right? But here we have some really interesting stuff happening in prophase 1, and that is that we've got our tetrad forming because of synapsis, and we have that crossing over thing happening. Hugely important stuff going on during prophase 1. Of course, then during metaphase 1, the chromosomes line up at the metaphase plate, but instead of being single file, like the word during mitosis, they're lining up 2 by 2 in metaphase 1 of meiosis. In mitosis, you separate those sister chromatids, you're done, and you're diploid. You're still 2n equals 6, right? It's just that the sister chromatids have separated. Here in meiosis, at the end of meiosis 1, you haploid, n equals 3. after meiosis 1 is done, you're going to further separate sister chromatids. And so you're still going to be haploid, n equals 3, and each of these cells, n equals 3. Haploid. Remember that it becomes haploid at the end of meiosis 1. Doing a quick comparison here between mitosis and meiosis, I believe this is our final comparison. When you think about DNA replication, it's happening before mitosis and it's happening before meiosis 1, but it is not happening before meiosis 2 because sister chromatids were already made in interphase before meiosis 1. There's no need to go through more DNA replication. The number of divisions is just the one. We do have that extra phase of prometaphase that we talk about in mitosis. It's not that it's not happening in meiosis, but we just don't talk about it. So we say that there are just 4 stages. That's going to happen twice. Prophase 1 and 2, metaphase 1 and 2, and so forth. We only go through that once for each mitotic division. Synapsis of homologous chromosomes, nope, doesn't happen in mitosis, but does happen in meiosis. It's when non-sister chromatids exchange genetic material at the chiasmata. The number of daughter cells, 2. The number of daughter cells, 4. And then, of course, the role of mitosis in multicellular animals, in fungi, and plants. They're used for growth, repair, and some species do use it for asexual reproduction, like I showed you with that hydra, or some plants do it that way. But it's growth, repair, and that's mostly it. For meiosis, we're talking about gametes and or forming spores. You might hear that term eventually being used when you move on to the next class, the following class in Biology 102. I'm not going to really hit those terms very strongly, so I give you permission to, just for now, ignore those stages of life for plants and fungi. So what do we need to know now? Well, genetic variation is a key term when we're thinking about sexual life cycles and how they contribute to evolution. You get a lot of genetic variation just from that process of it going through meiosis. And I have not even finished talking to you about some of that stuff, but I just wanted to take a step backwards and mention that mutation, remember mutation is change to DNA. Mutation is the original source of genetic variation, genetic diversity. Without mutation, you would not have all the beautiful and wonderful, amazing variations within a species or between species. Mutations actually create those different versions that we were calling alleles earlier. So different versions of an allele are caused by mutation. We tend to think mutation is a bad thing, but in reality, if it weren't for mutations, we would all just still be, single-celled bacteria living in a warm pond somewhere on the planet. Mutations are essential for evolution. But so is reshuffling of alleles during the sexual reproduction process. So chromosome behavior during meiosis and fertilization is actually responsible for most of the variation that exists in each generation. It's not mutation. Like I said, mutation, super important. If we didn't have mutation, we would all still be like bacteria. It's super important for evolution. But it's not super important. It's much, much, much more rare compared to all the variation that happens between parents and offspring. Offspring don't look exactly like parents in humans, right? They don't look exactly, we just look very, very similar. The reason we do look different is because of 3 main mechanisms. And those are independent assortment of chromosomes, which I have not gone into any details about yet. Crossing over, which I have gone into quite a bit of detail about, but we'll go into a little bit more. And this idea called random fertilization. So let me start talking about independent assortment. So let's talk about this independent assortment of chromosomes. And this happens during metaphase 1 of meiosis 1. And the idea is called independent assortment. How this works is in a very, very simple example here, let's say your 2n equals 4. So your diploid number is equal to 4, okay? 2n equals 4. 1, 2, 3, 4. And imagine that we've got the metaphase plate right here. So these are lined up on the metaphase plate. They can line up in a couple of different ways. Here's one way. You can see this. Actually, these two, this one here and this one here, are exactly the same. And stand by when I tell you that why these two are exactly the same, even though they look like they're flip-flop from each other. And that's because the end result would be exactly the same. So let's follow this pathway of thought with this one, and then we'll follow the exact same pathway as this one, and we'll see why they're the same. But why this one is actually quite different. So on this side of the metaphase plate, you have a long red chromosome and a short red chromosome. And on the other side, you have a long blue chromosome and a short blue chromosome. Right? You can think about these as paternal chromosomes and maternal chromosomes. You'd have to think about that as what you got from your mom or your dad. And when metaphase 1 finishes and you go all the way through meiosis 1, you would have a haploid cell. And in that haploid cell, you'd have one long blue and one short blue chromosome. Then we'd further go through that division, separating sister chromatids. Still, one long blue and one short blue. Over here, one long red and one short red. Sister chromatids separated, still one long red and one short red. Again, these two are functionally the same because in the end, when this one split, you'd still get one long blue and one short blue, and this would split into the long red and the short red. So these two are the same. But this is different because in the end, if you split this down the metaphase plate, you'd actually have a long red and a short blue going into a cell. That's very different from that. Yeah, it's like a mixy-matchy thing. So this is what I mean by independent assortment. You really have to look at sorry about that, just making sure I have a clean slate here. You really have to look at the end result. These could line up in the equator in two different ways. And that's only because we only have 2 pairs, right? We have a total of 4 chromosomes, 2 pairs. It could line up with the blue being on the same side of the equator and the red being on the same side of the equator, or it could be this mixy-matchy. And in the end, you end up having very different results here versus here. So that's the basic concept of independent assortment. And depending on the number of chromosome pairs you have, right? Depending on, like for that simple example where we only had 4 total chromosomes, where 2n equals 4, you can see that we can have 1, 2, 3, 4 different combinations of gametes. But for humans, that number ends up being 8 million possible combinations of chromosomes just from this idea of independent assortment. Again, the idea of independent assortment is how they line up on that metaphase plate is independent of however this one lines up. This one doesn't tell you how this one lines up, right? It can be mixy-matchy. So there are 8 million possible combinations of chromosomes. That's 2 raised to the 23rd power. The reason it's the 23rd power is because the haploid number for humans is 23. So again, just another way of looking at this. You could line up on the same sides, or you can line up on those opposite sides. And you would get 4 different combinations in the gametes. Now, how does crossing over add to genetic variation? Because just to back up a second, the whole point of describing this independent assortment was to really understand how you have variation in the next generation, right? So crossing over, that's some seriously cool stuff here. I mean, you're recombining information on the chromosomes that your mom and your dad gave you. You pass that information on to your offspring in a mixed up way, which is huge. In meiosis in humans, on average, you have between 1 and 3 crossing over events per chromosome. So it might happen once or twice or 3 times. That's wild. You can see it happening twice along this chromosome right here. Happens at that spot. There's a chiasmata there and there. And so you've got this very interesting mixy-matchy thing going on with these recombinant chromosomes. Crossing over contributes to genetic variation in the offspring by recombining the portions of DNA. It's not actually possible for us to calculate easily how much variation crossing over adds to the next generation. It's just a tremendous amount of variation. The last one that we were talking about is random fertilization. And with random fertilization, you get genetic variation because any of the 8 million different gametes that you can make can fuse with any of the 8 million different kinds of sperm that the other person in the partnership can make. And so those two fusion of 2 gametes, that's that you have to multiply things together. So the probability of 8 million times 8 million, it's actually 8.4 million. We were rounding earlier. That's okay. Is 70 trillion. 70 trillion different kinds of zygotes. Remember, zygotes are fertilized eggs. 70 trillion different combinations of zygotes just because of the idea of independent assortment times the number of ways that sperm and egg could combine. 70 trillion different possibilities that you could have been, and yet you are you. You got to be you. Again, random fertilization adds a huge amount of variation. But the thing that we can't calculate, and again, 70 trillion different kinds of pairs just from that. But we really have trouble adding in how much that crossing over thing adds. It's a lot. It's more than 70 trillion different possibilities. I can tell you that right now. So, that said, each zygote has a unique genetic identity, and we can take a look at the bigger picture and think, well, evolution, which we'll talk about later in the semester, evolution acts on genetic variation within a population. Natural selection is the accumulation of genetic variation depending on whatever is favored in a particular environment. So sexual reproduction contributes to that genetic variation on those 3 levels that we just talked about. And without that kind of genetic variation in a population, you would not be able to have evolution. Don't forget that mutation is the ultimate source of all variation, but the way that sexual reproduction mixes and matches whatever you got from your parents and passing those on to your offspring is pretty incredible. So that's all I've got for you for meiosis. I hope you enjoyed this lecture. It is very interesting. It's also very challenging. Let me know if you have any questions.